Good morning to one and all present here, and it's a pleasure to join the Society of Pediatric Urology. In this particular lecture, we will cover various aspects of systematic reviews and meta-analysis, what are the differences between them, how to start, what is a Prisma flowchart, heterogeneity, risk of bias, publication bias, etc., etc. Now, when we look at a particular research question, like is tip repair good for all distal hyperspadias? When we go through textbooks or literatures or libraries, we are overwhelmed with a lot of information and we do not know which article to decide uh, or, or go for that particular information, because there are so many articles which are in favor as well as against. So when we have a meta-analysis, for example, this one appeared in this year by Sile et al., who said that using an inlay graft during primary distal hyperspadias repair decreases the risk of stenosis. We are so happy that there is one study which combines all the evidence, or at least the recent evidence. So systematic reviews and meta-analysis thus form the top hierarchy of the evidence. The difference between traditional and conventional systematic reviews is traditional reviews are subjective with unclear selection. So they compare sometimes apples and oranges, whereas a systematic review is an objective review with a selection protocol. It narrows the question. The methods are very clearly defined. It's a comprehensive search to get all studies with a low quality control and a low bias. So systematic review is a review that is conducted according to clearly stated scientific research methods and is designed to minimize bias and errors inherent to traditional narrative reviews. A meta-analysis is a quantitative approach for combining the results of previous research to arrive at conclusions using statistical methods. So how do we do? The steps are like you develop a focused research question, you define inclusion and exclusion criteria, search the literature, select studies, assess the study quality, extract the data, analyze the data, then interpret the results and draw conclusions. So this is an article we published this year on gender dysphoria in BSD adults. And we had a very clear question: what is the prevalence of gender dysphoria? So we found that gender dysphoria is low in women with the CAH, a complete androgen insensitivity or complete gonadal dysgenesis, whereas it is high in women with 5-alpha reductase, etc. So we know that these children are better reared as male, as little babies. So formulating a review question is important, and it follows the same PICO criteria as what we use in randomized controlled trials. So in this meta-analysis of ours, which compared the outcomes of single stage versus two-stage hypospadias, we defined the population as a proximal hypospadias patients with a severe COGI. The intervention was like the urethral plate has been divided to release the COGI. So then we can compare whether single stage or two-stage is better in terms of the outcome, which is a complication in the form of a fistula, stenosis, glands dehiscence, various functional complications and cosmetic complications. So Prisma reporting refers to preferred reporting of systematic reviews and meta-analysis. And here we have to clearly spell out what are the search criteria we used, how many records we actually identified, how did we exclude certain articles, for what reason, and then we have to justify why we have included only 25% of the articles which we have actually identified. Now, searching for databases, you all know that Medline, Embase, and Cochrane databases are there. In addition, you can search some gray literature like a dissertation and conference proceedings. Only you should make sure that you have enough information, whatever you want in there, and you can check the existing systematic reviews in a similar data for ideas, and the cross citations can also be used, but only thing is it has to be thorough and complete. Selecting included studies, selecting potentially eligible abstracts is to assess the titles of the abstracts, and then you go to review the full text articles of the initial selections. then the full text is read thoroughly, and it is analyzed. It often requires a judgment about whether to include or exclude. So it probably needs more than one reviewer at times. Data extraction, you need to have a clear question. So what effect measures do you want to calculate? So what data do you need to get this? For example, if you want to look at a particular complication, you need to have the numerator and denominator. So if you don't have both of them, then you can't actually include that particular data. So you need to know how to extract the data. Then you also have to declare what is the heterogeneity, which is a variability in the population or interventions or outcomes. Heterogeneity is assessed by Cochrane StatisticQ and I2 statistics, and it can be low or substantial or high, but you have to declare it. So in this meta-analysis comprising laparoscopic and robotic pyeloplasty, the heterogeneity was actually low because all the data, this is the forest plot, which you all know by now. So they are all lying together, not all over the place. So they are pretty much within 90 to 100%. So that's where they are. And this is the pooled success rate of pyeloplasty, which is 97%, and the pooled success rate of robotic pyeloplasty was 94%. And there was no significant difference between these two success rates. So that is our conclusion. So each paper has to be assessed for a bias. The bias can be multiple types of bias, and I'm not going into the details of what are the types of bias, but we have to read each article for a particular bias. For example, in our paper on hyperspadias, we found that in this particular question, what was the cardiac correction type? Was it described uniformly? So that was the data 4, and then we had multiple judgment issues, like there was a lot of problems there, red lights. So that is a traffic light plot, which is a risk of bias done with a ROB-based tool. Now, publication bias refers to inclusion of studies with only positive or negative outcomes. So a funnel plot is a simple way of representing the publication bias. And so they should lie on either side, but not too much outside. I mean, many outliers means that there's a lot of heterogeneity. And if the studies are on only one side, then that shows that there is a publication bias. Doi plots and LFK index are other two measures to assess the publication bias. And I am not going to dwell into the exact details of this particular method of analysis. Now, you need to be aware of fixed effects model and random effects model. Fixed effects model assumes that the true effect of treatment is the same for every study, whereas the random effects model assumes that the true effect estimate for each study vary. So this is a study comparing grafted tip and tip repair, and these three articles compared head-to-head within the article. So they have made an odds ratio plot. This is the odds ratio plot, and this is for fistula, this is for glans dehiscence, and this is for overall complications. So in this article by Sile, the fistula rate favored grafted tip, glans dehiscence was not favoring anything. The overall complications again favored grafted tip. So this is performed with the help of a software called Revman meta-analysis software. Now, Luis Braga in the year 2011 came up with this article about methodological concerns and the quality issues of systematic reviews. Obviously, these were the meta-analysis done in the last decade before 2010. So they found that only one review described a full research strategy, and only 5 described the assessment of publication bias, and only half of them checked for heterogeneity. So according to Amstar criteria, they felt that only 3% of them were of good quality. But these things have changed, and in the current decade, the meta-analysis and systematic reviews are now describing all the data we need. And you all know about Cochrane database. It was started much earlier to make sure that the essential data are held up to date. And Prospero is an initiative where one can register the planned reviews and ongoing reviews and just to avoid duplication of systematic reviews. So in short, when you're overloaded with the information, life can be really taxing, Whereas if somebody can do a proper systematic review and meta-analysis and give only the essential data, life becomes so easy and you are thankful for that particular research work. So this is in short about systematic reviews and meta-analysis. I once again thank the organizers for giving me this opportunity.